All right, so now on to meiosis. A lot of times people think this is the hardest type of cell division, so I put my glasses on to make sure that we zero in on what we need to talk about here. So, I'm actually too far into this. So, in sexual reproduction, parents are going to be contributing different amounts of genetic, well, not different amounts, but different genetic contributions to an offspring. And so what happens are the gametes fuse together during fertilization and a new organism is formed. And that process leads to the growth of the individual through mitosis. And at some point in time, a set of tissue is set aside to become gonad tissue to start undergoing what we call gametogenesis. Gametogenesis is the formation of actual gametes, and that happens in the gonad tissue. In males, it'll be in the testes, and in females, it will be in the ovaries. So, lots of organisms produce sexually specific gametes. Sperm cells will always be the male cells. They're relatively small. They're pretty mobile. They travel around. Egg cells are very large relative to a sperm cell. They're very rich in nutrients. They have a lot of stored energy. They also have a large amount of mitochondria. Sperm do not bring mitochondria to the fertilization party. So one of the things to bear in mind in terms of mammalian species is that all the mitochondria that a mammal has, that even in birds as well, I guess you'd say vertebrate species, all the mitochondria come from mom, not from dad. Now, it turns out that plants actually also produce sperm and egg. It's just we call them something different. Sperm is called pollen, and eggs are called ovule. And so the pollen pollinates the plant, and you'll see this at the very end of this chapter. The pollen actually extends a pollen tube down the style of the female reproductive organ, and it delivers the nucleus for fertilization. Once that happens, a fruit forms around the fertilized egg, and that fruit is actually the ovary. And so in many species of plants, what we're actually eating as fruit is the fertilized ova. In humans, all gametes contain only 23 chromosomes. Because remember, mom and dad give an equal contribution to the resulting offspring. So all of our cells, with the exception of gametes, they all have 46 chromosomes. So how do you go from a cell that has 46 chromosomes to one that has 23? Well, that's meiosis. Meiosis is the formation of what we call haploid cells. Diploid cells have chromosomes for mom and dad. Haploid have half the number. And we're going to look at how that happens here in just a minute. So basically what happens in meiosis is the cell cycle kicks off normally. You go through G1, S, G2. In other words, the gamete cells, the pregamete cells, the stem cells that make ova and sperm, start producing new chromosomes. And you end up with sister chromatids. Then, when M phases hit, you go through what's called meiosis 1, and then you totally skip G1, S, and G2 again, and you go through meiosis 2. So what you have is G1, S, G2, prophase 1, prometaphase 1, metaphase 1, anaphase 1, telophase 1, going straight to prophase 2, metaphase 2, anaphase, and telophase 2, and then cytokinesis. So, prophase 1 itself is actually further subdivided into five different periods. The leptotene, the zygotene, picotene, diaplatine, and diakinesis. What's going on during all of this? These are important steps for what we call crossing over to occur. So during prophase 1, homologous pairs of sister chromatids, so X-shaped chromosome 1 and X-shaped chromosome 2, aren't sitting one on top of the other, like this, the way we saw during mitosis, I guess more like this, right? They're sitting next to each other, so that you literally have 4 chromosomes lining up. So in the end, they're able to swap information. But in order to get to this step, the leptotene is the first stage where replicated chromosomes condense and they start finding each other. So chromosome 1 from mom, chromatid pair, I guess I should say 1 from mom and chromatid pair from dad find each other. They form during the zygotene what we call the synaptonemal complex. This is where a piece of chromosome is then going to swap. Literally, between mom and dad's chromosomes, you're going to have a swapping of genetic information. And so that process further kind of gets oriented. That during the zygotene, you have this kind of zipper forming between the chromatid pairs. And by the picotene, you have open ends. During the diplotene, the chiasmata is very obvious, and that's where the crossing over is literally happening. They call the chromatid pairs a tetrad at this point because you have 1, 2, 3, 4, or tetra chromatids. Then at diakinesis, this is a step at the end of prophase 1 where you can see the completion of the swapping of terminal ends of individual chromosomes. Now, interestingly, you see this happening on the tip of the two interior chromatids. This can happen in between during a double crossing over event. This can be a very complex situation. During metaphase of meiosis 1, the sister chromatid pairs 1 and 1 line up on the imaginary metaphase plate. There's really never no plate there, or there's never a plate that you see. It just looks like a plate down the center of the cell. The metaphase plate is going to allow, just like we did in meiosis, metaphase of regular mitosis, is going to allow spindle microtubules to extend and contact the kinetochores, and eventually chromatid pairs are going to be separating. Now, this is different than in mitosis. In mitosis, you had X's going to single chromatids, okay? You had, let me find my camera. Chromosomes or sister chromatids separating to individuals. Sorry. Over here, you have chromatid pair and chromatid pair that were sitting together. They swapped information, and then pairs are separating here. They're going to migrate to opposite poles during anaphase 1, which we will see here. So the X shapes are migrating to opposite poles. And then during telophase and cytokinesis 1, you'll see the beginning of a new nuclear envelope forming around half the number of chromatid pairs. All right, and then a cleavage furrow forming. So here we started with 1, 2, 3 reds and 1, 2, 3 blues. 6 pairs. 6 pairs. 6 pairs. 1, 2, 3, 4, 5, 6. Again, 3 and 3, which makes 6. But now we're dividing, and you only have 3 here and 3 here. This first telophase and cytokinesis event is the end of meiosis 1, and we call this the reduction division. This is what's taking your diploid cell and turning it into a haploid cell. In other words, you have the two long chromatid pairs together here, and here they're separated. So chromosome 1 of dad, the chromatid pair from dad, came to this cell, and the chromatid pair from mom came to this cell. Chromatid pair 1 from mom, or 2, came here. Chromatid pair from 2 of dad came here, and so forth. Then, during meiosis 2, this looks just like mitosis. It's just you have half the number of chromosomes. So you'll have the spindle microtubules starting out, migrating to the opposite poles. The nuclear envelope dissolves. The spindle microtubules literally push the chromatids in single file, just like they did in mitosis. Then the chromatids, the X shapes, are pulled apart. And now each individual cell has only a single copy of chromosome 1, a single copy of chromosome 2, and a single copy of chromosome 3. And you have 1, 2, 3, 4 cells. So, the end of meiosis 1 is the reduction division. You go from haploid, diploid, oh my goodness, I am so sorry. You go from diploid to haploid. And then during the second division, meiosis 2, these cells remain haploid. You're just going from chromatid to chromosome. So, you started with chromatid pairs together. Chromatid pairs separate during meiosis 1. During meiosis 2, the chromatid pairs become individual chromosomes in 2 different cells for each. So you have 1, 2, 3, 4 cells being produced at the end of meiosis. So, I posted a video or a couple of videos identifying Meiosis and Mitosis. Definitely watch those. I think it's very helpful to see the process happening. We're very visually oriented organisms, and seeing a movie of how this very complicated process occurs is really helpful when you're preparing for an exam. Mitosis versus meiosis. Sounds a little bit like a discussion question. Might be coming your way. Mitosis gives you 2 diploid daughter cells. Meiosis gives you 4 haploid daughter cells. Mitosis gives you cells that are genetically identical, but meiosis gives you mixed up pairs. What am I talking about? Let's come back to this slide again. Remember we had crossing over happening during prometaphase right there? That little tip exchange mixed up information between mom and dad's chromosomes 1, and now each resulting cell, or 2 of the 4, have a new combination of chromosome 1. Another thing that happened here is that mom and dad's chromatids did not line up, all mom on one side, all dad on the other side. There was a mixing here. This is really interesting because what this does is it results in each new cell at the end of the division having a different amount of DNA from mom and dad. These 2 gametes have more mom's DNA. These 2 have more of dad's DNA. This is called homologous recombination. So in humans, spermatogenesis is the production of sperm. It happens in male organisms and it occurs in the testes. It takes diploid spermatogonial cells, allows them to mitotically produce 2 cells, and then they undergo meiosis. It's a little bit weird, all right? Here's a slide basically summing up how the process works. The structure of the sperm, long flagella. This is the only vertebrate cell that has a flagella. It has a sperm head. It's capped by an acrosome. The cap itself helps protect the sperm. And in human males, spermatogenesis continues for many, many years in adult males. Excuse me, hold on a second.